Hello, everyone. In this chapter, we are going to address genomes. As we go through the chapter, the first step we are going to make is to basically look into how we went from genetics to genomics. The second step is going to be on analyzing the genome content. How did we come to understand what is in our genome? And finally, we are going to take a look at our knowledge that we acquire and expand beyond just the knowledge of the genome. How do we use that knowledge, right? What comes next? Is there a better understanding that we have of some cellular processes that we addressed when it comes to genetics, including, for instance, the crossing overs and the mutations that occur in DNA? All right, with that being said, the study of genomes really can be traced back to when we were interested in why people die, why people get sick, what lies behind that sickness, what explains that. Is there something within us beyond infections that actually can explain our health issues, right? So a lot of postmortem analysis were done to actually try to understand what's going on with now what we know today as our genetic makeup. What's going on with it, right? So people started really looking into dead individuals, dead corpse, made analysis, and actually started really looking at the sequences of the genomes of these deceased people. About 300 deceased individuals were compared to actually see, what their genetic makeup was, and comparing that information with the database that already existed. So this was very important because it helped clinicians and researchers for preventing some diseases, detecting some potential, or the way we tried to explain some affections to associate them with the genetic makeup. But not only that, but the information gathered was basically also instrumental for treating specific diseases, right? So, because of the work done on people who were dead and from which sequence analysis was obtained, we were able to identify a number of things. For instance, that some cancers are associated with very specific genes, right? For instance, breast cancer, prostate cancer, to name only these two. They also showed that some genes that may affect human responses to certain medication actually were indeed involved in this. Now, there is a whole discipline today that we call pharmacogenetics. Pharmacogenetics, it's basically leading us to designing specific treatment for specific people because we don't all respond the same way to a treatment, to a particular medication. That's why, by the way, when we are designing new drugs for treatments, the way we go ahead with clinical trials becomes very important, because when we say that, well, this drug is good at treating, for instance, this form of cancer, the question we want to ask is, who were the people the drug was tested on, right? What is the diversity? Does it fairly represent the diversity in the humankind? Because we are obviously somewhat genetically different. Remember, several chapters back, we talked about single nucleotide polymorphism, right? When you consider single nucleotide polymorphism, this is going to account for our way of responding to treatment that is going to vary from one person to another. So this post-mortem analysis allowed to uncover this kind of information. All right, so how do we go from genetics to genomics? Well, the term genome actually was coined in 1920. It was coined in 1920 to refer to the set of chromosomes, right? And the genes that are found in these chromosomes. Now, the term genome actually refers to the content, the genetic content in a haploid cell. For instance, you consider a sperm cell. That sperm cell is haploid. An egg cell is haploid. So that set of all the chromosomes in a sperm cell represents a genome. In an egg cell represents a genome, which means that in a zygote, which is a fertilized cell, right? You have two genomes, one from mom, one from dad. In your somatic cells, your skin cells, your liver cells, your muscle cells, your nerve cells, you actually contain two genomes. One you receive from mom and one you receive from dad. Now, let me close that parenthesis. The term genomics actually did not occur until about 1986. And that term genomics focuses on the study of genomes, right? Being able to dig into the genomes and read the sequence of genes in a genome and be able to actually identify some specific sequences that are responsible for specific diseases and how these sequences can vary in a population from one group to another or even from one individual to another. All right, but how did we go about getting into genomics? Well, we go from genetics. Genetics, we looked at, our genetic makeup, the genotype, the phenotypes, and in general, from the phenotypes, we are going to ask, okay, well, what lies behind these different phenotypes, right? So that gets us to dig into the chromosomes, right? Now, there are different types of analysis that can be done when we start digging into the chromosomes. Now, the first step I would like to make is a type of study that we call linkage studies. This linkage studies allows us to not only associate a particular segment of the chromosome with a particular disease, but also the way the technique of analyzing the chromosome is set, we have an overlap that allows us to link the sequences. So really, there is two meanings to this. Now, as we consider a chromosome as the one you see here, right? That chromosome has many bands. And this bands allows us to map the chromosome, right? To know that, okay, well, in the chromosome, you have band, for instance, 22.3 that is located at a particular area of the chromosome, which is right here. You have the band 21.3 that is located on this area of the chromosome, right? You have band 13.0 that is located in this area of the chromosome. So we can actually label the chromosome in such way that we are going to know specifically where segments of clusters of genes are located on the chromosome. Now, the first type of map we are going to have that I'm going to talk about is the cytogenetic map. The cytogenetic map, cyto means cell, genetic gene. So basically, the cytogenetic map, it's a map of the structure of the chromosome. So you see a number of bands here, right? So this bands allows us to tell one chromosome apart from another chromosome, right? Now, these bands are going to occur in many types on a given chromosome. So in a cytogenetic map, this is going to allow from one band to another a spacing of at least 5,000 kilobases, right, apart. Now, when we zoom into the map, we are going to focus on a particular band, right? When we look at that particular band, at that point, we are going to use a unit that is actually the centimorgan, okay? So using the centimorgan units, we are going to actually look at the sequence of the genome in terms of hundreds of kilobases apart. At that point, we have a type of map that we call linkage map. And this linkage map allows us to have, all the little bands that we see here that are distinct one from another. So we are basically zooming into the cytogenetic map. Then in the linkage map, we can zoom some more, right? We can zoom some more, and there now we are going to see the genes, right? So this is one, this is one, this is one, this is one, and basically we are going to take a look at these genes, and these genes typically are going to allow us to describe what we call a physical map. That physical map is going to allow us to look at genes with tens of kilobases apart. Then we can zoom some more in the physical map. When we zoom some more in the physical map, now we can focus on a very specific gene, right? We can tell, okay, here is the sequence of the gene. The gene starts with, for instance, A, G, G, T, C, C, G, A, T, T, A, right? So that nucleotide sequence, it's actually what we call the sequence map. This is the closest you can zoom, right? It's like in a city, right? If you consider a Google map, right, you can obtain someone's address and be able to zoom on that address and know specifically where the person lives, right? You can actually see the house where the person lives. You can see the driveway and so on, right? Rather than just looking at the city, right? So this is basically the linkage studies that allows us to reach a point where we have a sequence map. By knowing the sequence map, we can actually tell when there are some discrepancies in some genes, right? We can basically be able to identify single nucleotide polymorphism in, for instance, that whole sequence that we have here. But let's take a look at other approaches. Then the next approach is basically the positional cloning. The positional cloning, it's a type of way of looking at the genome gene by gene. In a gene by gene approach, we are going to try to match up a gene with a specific disease. So we say that, for instance, okay, well, someone has cystic fibrosis. Okay, fair enough. The person has cystic fibrosis, but what gene is behind cystic fibrosis, right? So the researcher will start investigating and try to match the affection with a particular gene. So we are going from a phenotype, cystic fibrosis, frequent respiratory infections or digestive tract infections, to trying to find the causative agent, right? In this case, we are not going to really say causative agent. We're going to say the gene responsible for that condition. And then we are going to ask the following question. Wait, yes, there is this particular gene that is responsible for that condition, but question, which chromosome carries that gene? That's one. Two, which area of the chromosome is that gene located? Is it located on the long arm? Is it on the short arm? Where is it, right? So this approach has allowed researchers to characterize a number of diseases among humans, including the Duchenne muscular dystrophy, cystic fibrosis is another one, and the Huntington disease is another one, to name only these three. Now, the Human Genome Project actually took a while to develop, for the single reason that, well, technology was very limited. How do we get to understand the sequence of the whole genome? It was not easy from many perspectives. Now, these days, right, it takes a short period of time to sequence genomes, while before it took years to sequence genomes. But let me go back and tell you a little bit about the history, without too many details though. So the idea of trying to figure out the human genome emerged around 1980s. But really, the real work started in the 1990s. Now, because of how difficult it was to carry out that project, only wealthy people actually started developing some interest in knowing their genetic background, knowing if they had a chance to develop a particular kind of cancer, trying to develop treatments that were very specific to them and not a generalized treatment. So many motivations were behind the Human Genome Project, including also trying to understand the origins of an individual, right? About $3 billion were invested. It took about 15 years to actually achieve something that is deemed meaningful, right? And this whole effort, beside private people who really were able to inject good chunk of money, superstars, people who are extremely rich, but at the government level, there were also some efforts that were done, and particularly effort led by the Department of Energy and the National Institute of Health. The first draft of the human genome, was presented in 2001. However, it was considered finished in 2003 when I defended my PhD, by the way. Now, the whole Human Genome Project was not a one-person effort. It was an international collaborative effort that led to the understanding of what we have today. Now, let me tell you this. In spite of all the work that has been done, believe it or not, we are not done with the Human Genome Project, right? Now, don't get me wrong. We have a bunch of understanding, and we can associate specific genes with specific diseases, and we are even understanding these days that, well, in spite of all the discoveries and what we thought were particular conditions caused by one particular gene, we are realizing that, wait a minute, it's not that simple, right? It's sometimes a result of interaction between many genes, right? Or even that one particular gene can induce a bunch of alterations in the way our body functions. But let me close that parenthesis. So, what are the key events that led to discovering, really unraveling the human genome? So, there is this approach that we call Express Sequence Tag, or EST. The Express Sequence Tag focused on finding protein encoding genes. Now, how do you find proteins encoding genes? The easiest way was to simply isolate cells, and from these cells, we isolate messenger RNA. We know that the messenger RNA is a transcript of a gene, right? So, and we know that a messenger RNA is going to necessarily, technically, produce a protein. So from the messenger RNA, we did a backward approach, which is to produce a complementary DNA strand. That complementary DNA strand allows us now to produce the second strand of DNA, so that now you can have a double-stranded DNA molecule. And you can characterize that double-stranded DNA molecule to know specifically the sequence of nucleotide in that segment of DNA. Another approach, it's DNA microarrays. Now, maybe before I dive into DNA microarray, let me share with you the fact that when it comes to the production of cDNA using expressed sequence tag, well, remember that during gene expression in the human, we know that there is, after transcription, you have maturation of the RNA, right? There is splicing, there is even alternate splicing, right? This alternate splicing and even splicing itself, well, allows us to reach a point where we thought that a gene was just what the complementary DNA sequence was saying, so that we were only looking at exons, but really had no idea about introns, right? But the story after allowed us to actually be able to comprehend that, wait a minute, all genes are not just made up of exons. Some genes contains exons and introns, right? But let me close that parenthesis and come back to the DNA microarray. So in the DNA microarray, some small pieces of DNA were actually chopped from the chromosome and attached on a chip, what we ended up calling DNA chip. And that DNA chip, we were actually trying to match that DNA chip with a particular gene, right? Some probes, so that we were able to show what genes are expressed in a particular cell at a particular time. For instance, a gene that is a cancer gene, a cell that is a cancer cell compared to a cell that is a normal cell, right? How do we tell them apart, right? How do we tell which gene is expressed in a cancer cell, which gene is not expressed in a cancer cell? Well, microarray played a very important role there. So ultimately, we use microarray to sequence and assess gene expressions. What are the genes that are expressed in a particular condition in the cell? So then there was an approach by which the genetic information was cut into many pieces. These pieces were individually read, and there was an overlap of the different pieces. For instance, you have a piece of DNA that is randomly cut and read. So as this DNA is randomly cut and read, you have multiple sequences here, right? And these different sequences, the way we are going to build the nucleotide sequence is to see what overlaps with what. For instance, we have A, G, T, C, C, T. Here we have C, T, A, C, T. Here we have C, T, A, G. And here we have T, A, G, A, G, T. Now, when we actually look at how these sequences read and how they connect to each other, we can actually start aligning them like this, so that at the end of the day, we have a full sequence down here that reads C, T, A, G, T, C, C, T, A, G, C, T, A, C, T, right? Just by overlapping the sequences. So, how do we come with the sequence knowing that overlapping approach? Well, the first one of the approaches is to actually obtain a whole chromosome. That chromosome is going to be digested with restriction enzymes. These restriction enzymes are going to cut the chromosome wherever they have a matching restriction site, so that we're going to have many fragments like this. These many fragments actually are going to end up describing a long DNA sequence like the one you see here. And that long DNA sequence is called a quantig. Okay? It's called a quantig. Quantig stands for contiguous sequence. So these are sequences of DNA that are side by side with each other, so that we can have a long sequence. Now, these quantigs actually are inserted in a circular type of chromosome that is inserted itself in a bacterium. so that we have what we call bacterial artificial chromosome. These bacterial artificial chromosomes are going to be sequenced. And after sequencing, this is when we are going to know specifically that, okay, well, here is the sequence, right? We're going to know that specifically. And the good thing also is that, we are going to take the time to analyze each of the bacterium that is artificially modified, right? And see what it takes, what it contains. Now, here is the thing. That process takes a long time. I mean, years to actually execute. All right, other approaches, it's basically the whole genome. So right now, we are not focusing on one single chromosome. We are focusing on the whole genome. So we are going to do a shotgun approach. So just like we did earlier with restriction enzyme, we are going to use restriction enzymes here as well, right? These restriction enzymes are going to chop not one chromosome or a big piece of chromosome. No. It's going to be the entire genome, right? So that we have many fragments. And these many fragments are going to be organized in such a way that, well, we are going to see some of them belonging to a particular chromosome. But at this point, we don't know this yet, right? We don't know this yet. We really have to do what we call sequence tagged sites. This sequence tagged site is what is going to allow us to group the fragment and to say that these groups of fragments belong to this chromosome, and this group of fragments belong to this other chromosome, and so on and so forth. Now, that level of analysis also takes a long time. All right, so genome sequencing has gone from taking years to taking hours. Yes, to taking hours. Now, how is this possible? Because we have these days algorithms, right? That once you isolate a particular segment of DNA, right, you apply the enzymes that are needed, and the genome in a machine, so the machine right here is going to process all the information for us, right? The machine contains an algorithm that allows us to actually reach a point where we are seeing the overlapping areas so that we can deduct the sequence, right? So what this automated DNA sequencers are doing is to spit out of the machine because of a software, the nucleotide sequence. However, what the raw results do not tell you, it's what this nucleotide sequence mean in terms of exons and introns and genes, right? There is another level of analysis that will have to come, algorithms, right, that are going to tell that, well, when looking at this particular sequence, this looks like this is this gene. This looks like this is this other gene, so that we are going to have a clear idea as to the potential genes that are on the sequence that was just analyzed. However, while the sequencer and all these algorithms are going to give us an idea of the potential genes, we are only going to really know the validity of these answers only and if only we actually run analysis in a laboratory. Specifically, we are going to isolate a particular segment that is deemed to be a gene, right? We are going to see, is that sequence that is deemed to be a gene indeed a gene, right? Can we express it? And when we express it, what do we see, right? So this involves very complex analysis. That is the reason why, by the way, even though we have completed the nucleotide sequence of the human genome, there are still a lot of unknowns, because while there are a lot of putative genes of putative sequence that may account for something, we don't know for sure, right? And we still have to do that level of analysis. So what automated DNA sequences allow us to see is that there is about 0.07% of single nucleotide polymorphism, SNPs, that may affect our phenotypes. And this also allows us to have what we call reference genome. And these days we have improved technology that allows us to improve our sequencing ability. Actually, there is one particular sequencing technique these days that is called next generation sequencing. Next generation sequencing, for instance, is the type of sequencing that allowed us to actually be able to sequence the coronavirus, COVID-19, and the different strains of COVID-19s were actually identified using next generation sequencing approach. So in the sequencing approach here, just to move on again, we're going to have alignment of sequence that are derived from what we see from the sequencer, and then we can actually do a DNA microarray to confirm that indeed that sequence is correct. All right, so the annotation, once you obtain the nucleotide sequence, let me come back here. When you obtain the nucleotide sequence G, A, G, C, T, G, A, then what? Well, what does it mean in terms of gene, right? Now we have to do annotation, right? So this annotation, it's tedious, right? But it allows us to give a meaning to actually what we see from the algorithms, right? This allows us to, for instance, determine which gene is a normal gene, which gene is abnormal, what is the normal function, what is the abnormal function. It allows us to understand the mode of inheritance, right? Is that gene or the allele that we are looking at, is it recessive? Is it dominant, right? It allows us to determine the genotypes: heterozygote, homozygote, compound heterozygote. The frequency of variation in a particular population, we can be able to tell, right? And classifying genes are benign, likely benign, variant of uncertainty significance, the likely pathogenicity, right? Or pathogenic, right? So there is a bunch of information we can obtain here by doing this genetic analysis. Now, the limitations of genome sequencing are going to be that, well, even though we can have the alignment of nucleotides, we don't necessarily comprehend what it means in terms of health. And let me take you some examples. Now, some examples are many sequences that we thought were actually useless because the algorithms say that, well, they do not account for a particular protein, we are realizing that yes, they account for not proteins, but they have some functions in our health, right? When they are not there, we are not healthy. That's what we notice, right? But they have to be there, but we just don't know what they are for, right? We just know that when they are not there, well, we are not healthy, right? So there is still a lot that we are getting to understand. There is still a lot that we are getting to understand. So we cannot necessarily detect everything. For instance, one of the difficulties has been that some sequences are repeated multiple times. For instance, we can see segments of DNA that are CTA, 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 repeated many times. Now, in the traditional way of doing things, right, we can look at this multiple sequence of CTA as maybe a mistake. Maybe it's just actually one CTA sequence, and we would think that that's the correct one, but no, that's not the correct one, right? It's indeed multiple CTAs, and at the end of the day, it's end up having a particular meaning, right? Because from one individual to another individual, one person can have, for instance, 10 CTAs that align one behind another. Another person can have 8, another person can have 7, or another person can have 5, right? And we see that actually this end up affecting the way we all behave or do things or appreciate things. So, so what I just mentioned there, it's that, well, it's not easy to see the copy number variants. How about mitochondrial DNA? This is something that is not always captured by the typical sequencing approach. The uniparental disomy, okay, which is that you obtain 2 chromosomes, the same, from one of the parents, right? And we saw in the previous chapter that, well, a mom who was a carrier for cystic fibrosis end up having a daughter who actually has cystic fibrosis because, well, rather than receiving one of the copy from dad, no, the child receives the 2 copies from mom, right? And ended up developing cystic fibrosis. We have gene and gene environment interactions that we cannot comprehend just by looking at the genome and what we think the genome means, right? So today the concept of the genome, it's actually genome information that is very useful, obviously, but we don't know to which extent that knowledge that we just acquired is useful or not. So it's always a challenge to try to determine the medical value of this human genome project. So we are now trying to have a better understanding of the relationship between genomics and our health. So what else do I need to say? So DNA testing alone is not significant. It's not enough to actually pose the definite diagnosis. We have to look behind, because you have gene-gene interaction, gene-environment interactions. These are all things that are very important. But knowing the potential mutations that are present is going to be very important to look for ways to treat a particular affection, right? Or a particular condition. So the sequencing also allows us to understand the nuances we see in Mendel's law. We saw that now, while ideally one gene is responsible for a particular phenotype, we know today that actually this is not true, right? Because you just have many aspects that come into play. For instance, gene-gene interaction, right? You can see that one particular gene, while it is expressed because another gene is allowing that gene to be expressed, right? So it's not just simple to know the DNA sequence. And we see that the story is much more complex. Now, because of what we covered before, here we can see, for instance, that in a case of incomplete penetrance, it's a situation where the genotype alone does not always account for the phenotype, right? Other possibilities of variation, it's the variability in expressivity. The severity of a particular affection is going to vary from one individual to another. Epistasy, gene-gene interactions, genetic heterogeneity. This is where we have mutation in more than one gene causing a phenotype, and also obviously epigenetics, which is the influence of the environment. Now, let's take a look at things beyond just the sequence of DNA. Beyond the sequence of DNA, what genomics allows us to understand today, it's actually what is happening with crossing overs and the spontaneous mutations. We know that today, we know that crossing overs and de novo mutations or spontaneous mutations actually do not happen randomly. Before, we thought, well, crossing overs happens during prophase 1 of meiosis, okay? But we thought it was random, but actually we are finding out that it is not random, because there are many cases where we see a close association between some sequences of DNA and crossing over. That's one. Two, a close association between crossing overs and de novo mutations. And actually, there is even something that is happening with histones that actually sets the ground for crossing overs to happen and for mutations to happen. So that whole genetic diversity, it's not actually happening randomly, even though, right, it is random. Based on our understanding, it is random, but really, it doesn't happen randomly completely, right? So here we have crossing overs happening, right? Okay, in mom, in dad, right? We notice that, well, there are some histones that are modified here, that causes, based on if you are in mom and dad, on some mutations that are prone to occur in mom or in dad, and crossing overs that are prone to occur in mom or in dad. In any case, it's not the same thing. In mom and in dad, it's not the same thing. And Finally, looking at the genome in newborns is going to be tremendous in tackling some particular genetic affections, right? This allows us to actually come up with some new treatments. It allows us to actually prevent some situations from occurring. So, say for instance that a newborn has potentially a condition that would onset when they reach puberty, right? Now, with the sequencing, we can start thinking of ways to prevent that onset of the condition, right? So, these are things that can be done with genome and genomics, and which is actually transforming our health, not only right now, but really with all the analysis that are being done. A lot is still to be discovered, and the future can only be better in terms of our understanding and how to tackle some genetic affections in the future. Thank you for your attention.